Continuing on with the third and last part of our lecture, so we have the PubMed database. It's a search service from the National Library of Medicine that provides access to 18 million citations of Medline. Basically, it's a database for journal articles published in Medline. So, and other related databases. Actually, PubMed Central is the open access version for this PubMed. So, if you look at Entres at NCBI, you will find there um, two databases for the literature. We have PubMed PMC, PubMed Central, and there's also a bookshelf for this time for the books, published books or e-books on a wide variety of biotechnology, biology, and bioinformatics related topics, molecular biology related topics. So we also have the Entres. So Entres integrates scientific literature, DNA, and protein sequence databases, three-dimensional protein structure data, population study database, and assembling of complete genomes. Basically, Entres is the system that basically it's the database of databases. So it's the system that is basically when you search all databases in NCBI, you are actually using the Entres system, which searches the different databases under its belt for the related information. So for the PubMed, it's the literature component of Entres. So let's look at some practical applications or practical use of this one. So access to sequences, Entres Gene at NCBI. So Entres Gene is a great starting point. It collects key information on its gene or protein from major databases and covers all major organisms. So the RefSec, the system. RefSec is a system. It provides a curated, or RefSec is a project that provides curated information. Basically, entries that are curated by RefSec, you are sure that it's reliable and you can use that for your studies. So RefSec provides a curated optimal accession number for each data. So it's either a DNA or a protein. So we can, for practical purpose, actually you can do this on your own. So, and I actually have provided supplemental videos on how to actually access or use an NCBI database. So if you look at the web page of the home page or the landing page of your NCBI, you can just type beta globin and it will give you, this is the table of the different databases that, since you did not specify a database previously, it searched all databases under its management and how many entries you have. So for example, in PubMed Central, in terms of beta globin, it finds 15,047 entries. We have the PubMed for 8,403. And then some other sites, site search 31, so related sites. We have the books and then the OMIM databases. These are the entries in there. So if you want to know about a particular gene, so take note here, gene and nucleotide. So there might be some sense of confusion. We are looking here for a curated gene. So when we say nucleotide, they can be fragment of a gene, not the whole complete gene. So that is why when you look at this one, you only have 108 entries and we have 2,805 entries of beta globin. So nucleotide is basically more of a catch-all for DNA fragments, and other nucleic acid sequences that might have. Okay, so you want a beta globin gene. So first, when you are using this NCBI, you should have a sense of knowing. You should know what you really want to find out. You have a concrete sense of what exactly are you looking for. Otherwise, you would get lost. So you want to look at the gene. So look at the gene. So just click on the link. So it would give you the entries where it finds hemoglobin gene. So the entrance gene is the header for the official symbol HBB4. We have the official symbol HBB. It's human hemoglobin beta, and then some options for the limits, because this one, it gives us the basically there are lots of entries here, so uh, it can be from other species of human beta globins. So genes, genomes, SNP, gene view, invariation view. So there are some filters you can use in your search. So there are limits options here. So when you click on, when you click on a certain entry, so it will give you the uh, this is the GenBank entry. So this is the full report. So the display settings, it allows you to look at this is the GenBank entry. If you want to look it in a FASTA format, you can just look it as a FASTA format. So there are also some important links to other databases that reference this particular gene. So you have this summary page of your gene. Now, if you want to have a different display view, so this is the same entry, but if you notice here, the instead of summary, it becomes the GenPep. So this is another view for this gene view. So you have the locus, which where in the chromosome you would find it. You have what is it about the decision, and then accession number, NP. So that means it's a reference gene. It has been curated. So NRES protein, features of protein and sequence in a one-letter amino acid code. So this is, if you look at, if you click on protein and you look at the protein entry, so this is the protein sequence of the HBB in a single letter amino acid. So at least familiarize yourselves in the single letter amino acid sequence. They prefer this one instead of the three-letter code because it saves memory. So you just need to have one letter versus three letters, and for protein sequences that are much longer, it would be difficult to actually store this information. So you want it compact to be able to store it properly. So this is the one letter amino acid code. So this is, here is the actual sequence. So these are some additional information on the database entry. Okay, so again, familiarize yourself in the one letter amino acid. It's actually quite intuitive. You would immediately know, for example, alanine, A, ALA. So arginine becomes R because there's already letter A, and you have the next letter is arginine. So when we look, and even if we say arginine, arginine, so it becomes R. For asparagine, again, A is already taken. Why not S? We have a different amino acid that has S. It's serine. So it's asparagine. So N. So it becomes N. And then for aspartic acid, now here is quite a bit um, curious story. So aspartic acid, there already is an A, there already is an S, there already is a P. P is for proline. Now, where in this spelling is the letter D? So again, if you look at each individual letters, all of them are already represented properly. So when we speak aspartic acid, some pronunciation becomes in the tick, aspartic acid, dick. So it becomes somewhat like D because the way we pronounce it, aspartic. So it sounds like letter D, so it becomes letter D. Then you have the cysteine, again, letter C. Glutamic acid. Okay, here is glutamic acid. It's another curious story. So again, the same reason, G, L, and U, they are all taken, or at least for the letter U, you cannot use letter U because U is the uracil in the mRNA sequence. You can't use that. So again, we have G, we have L. G is for glycine. Okay. Now, L is, we have leucine. So what do we know? So when because before the one letter code, we have the three letter codes for the amino acids. When we talk about glutamic acids, it's already glue. So it's basically sounds like glue. I mean the glue for handicraft items. So they just add a letter E so that it becomes just glue. So it's glue. Glutamic acid is the glue without the E for the three-letter abbreviation. So for the one letter, let's just add the missing letter E at the end. So it becomes glue. So it is E. And then for glutamine, sounds like cutamine. Glutamine, cutamine. So it's Q. Glycine is G. histidine is H, isoleucine is I. So these are intuitive. Same goes for leucine. Now lysine is another curious tale. So lysine, you cannot have L, you cannot have Y, you cannot have S. And then the other letters are again already taken. So what should be the letter? So how about if it can be letter M, what is the next letter in the alphabet? M. But M is already methionine. What is the letter before L? It's K. And that is why it becomes letter K for its one letter abbreviation, because nothing else, basically they run out of ideas perhaps, so they just look at the alphabet. It cannot be M, so that's what is the letter before L. It's K, and that's why it became K. Then for phenylalanine, I guess the reasoning here is, again, sounds like the PH sounds is pronounced as an F, so it's phenylalanine F. Proline is P, proline, so S for serine, threonine. Now, how about tryptophan? Tryptophan is W because the structure of a tryptophan is a double ring structure. So it's a double ring, so its abbreviation becomes W. So that's it. And then the rest, tyrosine, we cannot use T, but the next letter is Y, and it's tyrosine. When we pronounce it, it's not tyrosine, it's tyrosine, so Y. So it's Y. And then again, valine. Lastly, for the valine is letter V. So basically, those are the one letter abbreviation. So it helps if you know uh, the story behind some of the counterintuitive letters, like the lysine, K for lysine. So it helps you remember. Okay, so the interest protein, uh, for the protein entry of the hemoglobin, so this is a Homo sapiens protein. So you can look at this graphics. This is the graphics format view, so you would be able to see graphically the shape of the protein. Now, in the FASTA, if you toggle the FASTA format, so you have this FASTA format. This is the FASTA. So we would be able to know what the FASTA format is because it starts with greater than sign. So, and then the ID here. So this is the FASTA format. We usually use FASTA formats uh, in doing analysis on the sequences, because the bioinformatics tools uses FASTA format because it's shorter, it's more compact, and it basically tells you what this the only signal that, for example, if it's a computer algorithm, it reads greater than sign as the line with the greater than sign means it's the ID of the gene that would be below it. So this is the sequence. So it's a faster, more compact way. And it's the one that we use, the format that we use when we do a sequence analysis. So take note of this type of format. Okay, so FASTA is a one file format. So aside from the FASTA, we have FASTQ or FASTQ with quality scores. This mostly when when you are doing analysis on sequences. So you enter it with a FASTA, it returns you with a FASTQ format, because that means your sequence have already have scoring. So we have the BAM, we have the SAM, sequence alignment map, and then we have the VCF. So these are the different formats. Take note of the formats that your software requires when you are using bioinformatics softwares for analysis. Take note of the format it wants, it requires as the input, and take note of the output file format. So because sometimes when you input, for example, FASTA, it returns you with a FASTM or a FASTQ. And then you need to be able to visualize it. So you need to have usually a visualizer tool, another tool that must be able to read that format and allow you to visualize it. So there are also some Excel formats. That .txt is another that text and that CSV. So these are more universal, but they are also used sometimes in bioinformatics databases. So again, be careful of this file formats at the end of your um, files. Okay, and aside from the interest tools, you have the BLAST. So we will be discussing BLAST later on in the class as we proceed in the module. So it's the basic local alignment search tools. This NCBI sequence similarity tool. So if you want to know if your query sequence of similarities in the database, so you can use BLAST. So it is a similarity of it's used to compare sequence similarities. So, and then we have OMI, Online Mendelian Inheritance of Man. It's a catalog of the human genes and genetic disorders. So, this is a database for genetic disorders. So, if you want to study about genetic disorders, you can start at the OMIM database. So, it contains experimental information, curated and not so much curated information on studies for the human genes and genetic disorders, and it also has links to your some PubMed articles and data. And then for the taxonomy database or a taxonomy website, it also has its own basically it says it's a database, but it's more of a sub database. So it contains taxonomy information. So if you want to have taxonomic information on certain cultures of bacteria, so, or even eukaryotes, you can look at the taxonomy. Although this is not so much as well used as GenBank, but you can look this taxonomy website for NCBI. And then the structure. This structure site maintains a molecular modeling database, which is a database of the macromolecular 3D structures for your site. So most of the entries here are PDB, protein data bank information. So these are structural information of the proteins and other um, molecular structures. So as long as it's a three-dimensional structure. So we have resources at the structure database or the structure site. We have PDBs. So this is a taxonomy site. It's the CN3D, which is the viewer, the basic CN3D viewer. Then we have the vector alignment search tools, where you can compare it based on the structure, not based on the sequence. So that's the VAST tool. So these are the structures. And again, so we have these large different tools, different sites, different databases. So in order to properly maximize the use of this one, first you must be able to, again, the very first thing that you need to know is you must be able to know what you are looking for, because there's a lot of information here and it's easy to get confused, it's easy to get lost if you don't really know what you are looking for and why you want to know that. What exactly are you looking at? What exactly is the objective? Be very, very specific what you want to know, because otherwise you would just get lost, because it's a very, very vast and very varied information. So these are the sites, the links to the sites of these databases. Okay, so that's it for this lectures and series of videos for this particular lectures. So please see the activity posted in our Blackboard. For the activity for this week, it's an individual activity. Okay.